Hi, this is Dr. Cassandra Quave, and in today's lecture we'll be covering Chapter 9 from your textbook, discussing an introduction to taxonomy covering the bacteria. Now, in science, being able to accurately use standardized names is essential. Because we're faced with a great number and diversity of organisms, biologists use the characteristics of different organisms to describe specific forms of life and to identify new ones. The grouping of related organisms together is the basis of classification. The most obvious reasons for classification are to establish the criteria for identifying organisms, to arrange related organisms into groups, and to provide important information on how organisms evolved. Taxonomy can be defined as the science of classification, and this actually helps to provide an orderly basis for the naming of organisms and for placing organisms into a category or taxon. Another important aspect of taxonomy is that it makes use of and makes sense of the fundamental concepts of unity and diversity among living things. So, organisms classified in any particular group have certain common characteristics, meaning that they have unity with respect to these characteristics. A basic principle of taxonomy is that members of higher level groups share fewer characteristics than those of lower level groups. Now let's talk about binomial nomenclature. So in the 18th century, there was a Swedish botanist named Carolus Linnaeus, and he's the one that's credited with founding the science of taxonomy. He invented binomial nomenclature, or a system which is a system that is still in use today to name all living things. Binomial means that this is a two-name system. And the first name designates what is known as the genus of the organism. The second name is the specific epithet, or the species. Together, the genus and that specific epithet identify the species to which the organism belongs. The name of an organism can often tell us something about it, such as its shape, where it may be found, what nutrients it uses, who discovered it, or even what disease it causes. And you can see some examples of names and their meanings in Table 9.1 in your textbook. Now, as a rule, members of a species cannot be divided into significantly different groups on the basis of a specific characteristic, but there are exceptions to this rule. In fact, sometimes members of a species are divided on the basis of small but permanent genetic differences, such as a need for a particular nutrient, resistance to a particular antibiotic, or the presence of a particular antigen. When an organism in one pure culture of a species differs from the organism in another pure culture of the same species, the organisms in each culture are designated as strains. A strain is a subgroup of a species with one or more characteristics that distinguishes it from other subgroups of the same species. Here you can see figure 9.2 in your textbook, and this shows us how a human, a dog, a wolf, and a bacterium may be classified following this system. Again, at the beginning you can see that you have the kingdom, then the division or phylum, the subphylum, the class, order, family, genus, specific epithet, and then subspecies or strains. Take a moment to look at this figure to compare how each of these different types of organisms is similar and yet different, but all of which are classified using the same system. Now, biologists often use taxonomic keys to help them identify organisms according to their characteristics. The most common key is known as the dichotomous key, which has paired statements describing characteristics of an organism. And here you can see an example from figure 9.3 and 9.4 of your textbook that shows you a dichotomous key for bacteria. To begin, for example, you might start with an organism by identifying it through a gram stain. Is it gram-positive or not gram-positive? And this will tell you where to go. Let's pretend that your organism has stained as gram-positive. We'll go then to step 2. What is the shape of your organism? Are they spherical or not spherical? Let's say that they're not spherical. This would send us to step 4. Are they rod-shaped or not rod-shaped? I'm going to say that they are rod-shaped, and so the answer here would be a gram-positive bacillus is the kind of organism that you may be looking at. So dichotomous keys can be important tools in helping us, again, to determine what types of bacteria you may be looking at or studying in the laboratory. Now, among the aims of a taxonomic system is to organize knowledge about living things and establish standard names for organisms so that we can communicate about them. Ideally, we would like to classify organisms according to their phylogenetic or evolutionary relationships, but this is not always an easy process. And this is because evolution occurs continuously and at a relatively rapid rate in microorganisms. It's far more important to have a taxonomic system that reflects our current knowledge than to have a system that never changes. Now we're going to discuss some of the different kingdom systems of classification. Now, before we discuss the 5-kingdom classification system and how it applies to microorganisms, we must first emphasize that all living organisms, regardless of the kingdom to which they are assigned, display certain characteristics that define the unity of life. All organisms are composed of cells, and all carry out certain functions. The cell is the basic structural and functional unit of all living things. Now, this is different from viruses, for example. Viruses are not cells, and this is one reason that they are not considered to be living organisms. All cells are bounded by a cell or plasma membrane, and they also carry genetic information in the form of DNA, and they have ribosomes where proteins are made. All cells also contain the same kinds of organic compounds, such as proteins, lipids, nucleic acids, and carbohydrates. They also selectively transport material between their cytoplasm and their environment. So, although organisms may be classified in very different taxonomic groups, the cells all share many similarities in both structure and function. Now, as we go into these different systems, I wanted to note also that no single classification system is completely accepted by all biologists. The 5-kingdom system, though, is one of the most widely accepted. A major advantage of this system is the clarity with which it deals with microbes. It places all prokaryotes, or microorganisms that lack a cell nucleus, in the kingdom Monera, and you can see this here in the diagram. It places most unicellular eukaryotes, organisms whose cells contain a distinct nucleus, in the kingdom Protista. And you can find more details on the 5-kingdom classification system in Table 9.2 on page 246 of your textbook. And here you can see the differences and the similarities between each of these different groups. Now let's discuss the kingdom Monera in more detail. The kingdom Monera is also called the kingdom Prokaryote. It consists of all prokaryotic organisms, including the eubacteria, the cyanobacteria, and the archaeobacteria. All monerans are unicellular. They also lack a true nuclei and generally lack membrane-enclosed organelles. Their DNA has little or no protein associated with it, and reproduction in this kingdom occurs mainly through binary fission. Now, of all the monerans, the eubacteria are of greatest concern in the health sciences, and these will be discussed in greater detail in following chapters. The cyanobacteria, which were also formerly known as the blue-green algae, are also of special importance in the balance of nature. These are photosynthetic, typically unicellular organisms, although cells may be connected to form thread-like filaments. Because these are autotrophs, cyanobacteria do not invade other organisms, so they pose no health threat to humans, except for the toxins that they may release into the water. Now, the archaea that survive today are actually primitive prokaryotes that have adapted to extreme environments. Examples of this could include the methanogens that reduce carbon-containing compounds to the gas methane. Extreme halophiles are also grouped in this category, and these again live in excessively salty environments, whereas the thermoacidophiles live in hot acidic environments, such as volcanic vents in the ocean floor. Now let's discuss the kingdom Protista. Although the modern protist group is very diverse, it contains fewer kinds of organisms than when first discovered. All of the organisms that are now included in the kingdom Protista are eukaryotic. Most are unicellular, but some can be organized into colonies. Protists have a true membrane-enclosed nucleus and organelles within their cytoplasm, as do other eukaryotes. Many of the protists live in fresh water, but some can live in seawater and a few live in soil. Another important point to mention is that protists do not develop from an embryo as plants and animals do, and they do not develop from distinctive spores as fungi do. But among the protists are the algae, which resemble plants, the protozoa, which resemble animals, and the euglenoids, which have both plant and animal characteristics. So the protists are of great interest to health scientists. The ones that are of most interest, again, are those that can cause disease. Now, the kingdom fungi includes mostly multicellular and some unicellular organisms. Fungi obtain nutrients solely by absorption of organic matter from dead organisms. So even when they invade living tissues, the fungi typically kill the cells and then absorb the nutrients from them. Although fungi do share some common characteristics with plants, their structures are much simpler in organization than true leaves or stems. Fungi form from spores, but they do not form seeds. Many fungi pose no threat to other living things, but some can attack plants and animals and even humans. Others can include things like yeast and mushrooms, which are important as foods or in food production. Now, the macroscopic green plants are placed in the kingdom Plantae. Most plants live on land and contain chlorophyll and organelles called chloroplasts. Plants are of interest to microbiologists because some can contain medicinal substances. A good example of this could be quinine from the cinchona tree, which was used to treat malarial infections in the past. Other plants of interest could include things like salix, which was one of the leads to the discovery of aspirin. Now, the kingdom Animalia, on the other hand, includes all animals derived from zygotes. And again, to define a zygote, it's a cell formed by the union of two gametes, such as an egg and a sperm. Although nearly all members of this kingdom are macroscopic and aren't really of concern to microbiologists, several groups of animals live in or on other organisms, and some can even serve as carriers of microorganisms. For example, certain helminths are parasitic in humans and other animals. Helminths can include things like flukes, tapeworms, and roundworms, which live inside the body of their hosts. Certain arthropods can also live on the surface of their hosts, and some can spread disease. Examples of this would include things like ticks, mites, lice, and fleas. Mosquitoes can also spread infectious microorganisms from their bodies to their host. Now we're going to move on to discuss this concept of the three-domain classification system. Now, in 1990, a new taxonomic category was suggested, and it was called the domain, to be erected above the level of kingdom. This suggestion was based on comparative studies of prokaryotes and eukaryotes at the molecular level and on their probable evolutionary relationships. Now, you have the standard view was that there was a universal common ancestor that first split into bacteria and archaea and then the eukarya, which branched off from the archaea. A second view, on the other hand, held that all three domains arose simultaneously from a pool of common ancestors that were able to exchange genes with one another. A third view sought to explain how so many genes are present in eukarya but lacking in archaea and bacteria, and this theory postulated that the existence of a fourth domain that directly contributed genes to the eukarya and then became extinct. Now, to discuss a little bit more about some of the archaea, you can see here a picture of archaea bacteria, which are, here's an example of an extremophile that can exploit this unusual habitat of a black smoker vent. This is a vent found at the deep part of the ocean, where hot sulfurous volcanic gases are released from the Earth's interior, and archaea bacteria can survive in one of the most extreme environments known. This particular vent is located in the mid-Atlantic ridge, about 3,100 meters below sea level, and it's under an incredible pressure of water and at a temperature of 360 degrees Celsius. And what's interesting here is that the bacteria that live there are able to survive these harsh environments and actually obtain their energy from sulfur compounds. Here you can see figure 9.11 from your textbook. This is from page 249. And this kind of helps to explain the changing system of classification, showing that systems of classification have progressed from the simple Linnaean model of 2 kingdoms to the current 5 kingdom, and again the 3 domain arrangement. So at the top you see this prokaryote and eukaryote two kingdom system down to the 5 kingdom system involving the kingdoms of Monera, Protista, Fungi, Plantae, and Animalia, and the last one that I mentioned, which is the 3 domain system of Protista, plants, and animals. Now, in this 3 domain system, the domain eukarya contains all those kingdoms of eukaryotic organisms, the animals, the plants, the fungi, and the protists. And the traditional kingdom Monera has been divided into 2 domains, the domain bacteria and the domain archaea. Now, as I mentioned before, there are some different theories about the 3 domains. The standard view is that the universal ancestor, again, split into bacteria and archaea and the eukarya, then branched off from the archaea. However, an emerging view is that all 3 branches evolved independently from the same pool of genes. And again, there's a third view that there was indeed a 4th branch that's now lost that once also contributed genes to the eukarya. And again, these are all just theories that scientists have come up with in an attempt to understand and explain the relationships between different groups of organisms. Now here you can see a figure from page 252 of your book illustrating the shrub of life. Now the shrub of life, while it's still tree-like at the top, the bottom does not come from a single trunk that arises from a single common ancestor. Instead, it suggests that life probably arose from a large population of many different primitive cells that eventually exchanged and shared their genes via lateral gene transfer. And if you recall, lateral gene transfer is a topic that we've covered in previous chapters. These links are shown by somewhat randomly placed across cross branches as the specific sequence of most transfers is unknown still today. However, it does show that the eukaryotes obtain chloroplasts and mitochondria from bacteria. Here you can see a better illustration of lateral gene transfer again. The colored lines arising from an assortment of different ancestral cells indicates lateral transfer of genes from one cell type to another. As the genes from varying sources are combined, they rise to give to new types of cell lines that have multiple origins of ancestry. Now, I mentioned earlier in the chapter that viruses are not considered to be living things because they lack the structure of a cell. But viruses are, however, incredibly important to the field of microbiology. Viruses are acellular infectious agents that are smaller than cells. They contain nucleic acid, which may be in the form of DNA or RNA, and are coated with a protein. These have not been assigned to a kingdom. In fact, they only display a few characteristics associated with living organisms. Initially, viruses were classified according to the host that they invaded and by the diseases that they caused. However, as more was learned about viruses, the early concept of one virus, one disease has been shown to be invalid for many viruses. Instead, today, viruses are classified by chemical and physical characteristics, such as the type and arrangement of their nucleic acids, their shape being cubical or tubular, and the symmetry of the protein coat that surrounds the nucleic acid, and the presence or absence of such things as a membrane covering, called an envelope, enzymes, tail structures, or lipids. And here you can see an example of how some different viruses may be classified using the type of DNA, the type of nucleic acid they contain, if it's DNA or RNA, whether or not they're enveloped, and you can see how this is broken down. Now, the study of viruses or virology is extremely important in microbiology for two reasons. First is that virology is a recognized branch of microbiology, and techniques to study viruses are derived from microbiological techniques. And secondly, viruses are of concern to health scientists because they can cause many diseases in humans, other plants, animals, and even in microorganisms. Now let's go on to speak about taxonomic methods. Now, many biologists are interested in how living things evolved and how they are related to one another. In fact, most people have some curiosity about how life originated and gave rise to diverse assortments of living things that we see today. Here we'll discuss some of the different types of taxonomic methods. Now, we know that the taxonomy of most eukaryotes is based on morphology, such as structural characteristics of living organisms, genetic features, and on knowledge of their evolutionary relationship from fossil records. However, morphology and fossil records provide little information about prokaryotes. For one thing, prokaryotes have left very few fossil records. However, there have been some rocks that contain fossils of individual cells of cyanobacteria that have been discovered, but they've really failed to reveal much information about the organisms. So because morphology and evolution are of little use in classifying prokaryotes, we use metabolic reactions, genetic relatedness, and other specialized properties to determine their degree of relationship. Let's start with a discussion of numerical taxonomy. This is based on the idea that increasing the number of characteristics of organisms that we observe, increases the accuracy with which we can detect similarities among them. If the characteristics are genetically determined, the more characteristics that two organisms share, the closer their evolutionary relationship should be. Although the idea of numerical taxonomy was developed before computers were available, computers now allow us to compare large numbers of organisms quite rapidly and according to many different characteristics. Here you can see in your figure an illustration of a probable evolutionary tree that was arrived at using numerical taxonomy. So here, organisms are compared on the basis of a large number of characteristics, and those organisms with a high proportion of characteristics in common are presumed to be closely related. So if you place your finger on number 1 in the image, or in the figure, and trace the line out to a specific organism, such as B or F, each number that your finger passes over en route is a characteristic possessed by that organism. Now, with regards to genetic homology, or the similarity of DNA among organisms, this really became an important tool following the discovery of the structure of DNA, in that it allowed us to study taxonomic relationships and the evolution of eukaryotes, again using the similar tests that can assess the similarity of DNA among organisms. One key tool in this process is looking at base composition. Organisms can be grouped by comparing the relative percentages of bases present in their DNA of their cells. Again, DNA contains 4 bases: adenine, thymine, guanine, and cytosine. We know also that base pairing occurs only between adenine and thymine and between guanine and cytosine. By making base comparisons, you can determine the total amount of guanine and cytosine in a sample of DNA and express it as a percentage of the total DNA. And by subtracting this percentage from 100, you can also get the percentage of total adenine and thymine in the sample. So we've found that using studies of base composition have shown that the guanine-cytosine content varies from 23 to 75% in bacteria. These studies have also revealed that certain species of bacteria, like Clostridium and Staphylococcus, have very similar DNA compositions, but Pseudomonas has a very different DNA composition. Thus, we can say that if the percentages in 2 organisms are quite different, they are not likely to be closely related. Now, automated equipment for identifying the base sequences in DNA or RNA is also available now at a reasonable cost. Using PCR techniques or polymerase chain reaction techniques and a DNA synthesizer, we can also use a large number of probes or single-stranded DNA fragments that have sequences complementary to those being sought. When the probe finds its target DNA, it will bind to it and not wash off when rinsed, and then the specimen can be examined for fluorescing dye on it or for radioactivity. The presence or absence of the unique DNA sequence can also help in the identification of the specimen. Now, protein profiling and amino acid sequences can also be useful in this process. We know that every protein molecule is made up of a specific sequence of amino acids and has a particular shape with an assortment of surface charges. Modern lab methods can allow cells or organisms to be compared according to the properties of these proteins. A protein profile is a laboratory-prepared pattern of the proteins found in a cell. Because a cell's proteins are the product of its genes, the cells of each species synthesizes a unique array of proteins, as distinctive as a fingerprint is for humans. Thus, analysis of these profiles of different bacterial species can also provide a reasonable basis for comparison. Now let's discuss DNA hybridization. Here you can see a figure that shows that strands of DNA are separated, and individual strands from 2 different organisms are allowed to anneal or join by hydrogen bonding at sites where there are many complementary base pairs. The degree of annealing will actually help to reflect the degree of relatedness between the organisms, based on the assumption that annealing takes place only where genes or parts of genes are identical. A high degree of homology or similarity, again, exists when both organisms have long initial identical sequences of bases. Close DNA homology indicates that 2 organisms are closely related and that they probably evolved from a common ancestor. A small degree of homology indicates that the organisms are not very closely related. Here you can see a figure that illustrates this concept of protein profiling that we discussed before. And what it also shows is the use of something known as polyacrylamide gel electrophoresis, or PAGE method. This method can be used to separate proteins on the basis of their molecular size. So in this method, samples of protein obtained from lice or cells that are broken open are dissolved in a detergent and poured into wells of a thin slab of polyacrylamide gel. The slab is then put into a buffer chamber, shown here, and an electrical current is then passed through the gel for a period of time. The current causes the protein molecules to migrate to the opposite end of the gel. Large protein molecules migrate more slowly than smaller ones do. After the smallest proteins have migrated, the current is turned off and the gel slab is removed. It is then stained so that various proteins can show up as separate stained bands in the gel slab. Now, bands in the same location and profiles from different kinds of cells can indicate that the same protein is present in the different cells. Also, determination of amino acid sequences in proteins can also identify similarities and differences among the organisms. The proteins of an organism are determined directly by the information in that organism's DNA. So both protein profiles and the determination of amino acid sequences are as significant as a measure of the relatedness of all organisms as are DNA homologies. Here are some other techniques that can be useful in testing for evolutionary relationships of organisms. One is that of ribosomal properties. So we know that ribosomes serve as a site of protein synthesis in both prokaryotic and eukaryotic cells. RNA in ribosomes can be separated into several types according to the size of the RNA units. A particular RNA unit, the 16S rRNA component, has proven especially useful in studying evolutionary relationships. Immunological reactions can also be useful in identifying and studying surface structures and the composition of microorganisms. One highly specific and sensitive technique involves proteins called monoclonal antibodies. These can be created so that they will bind to a specific protein, usually one that is found on the cell's surface. If the antibodies bind to the surfaces of more than one kind of organism, the organisms have that protein in common. Now, another important technique is that of phage typing, and this involves the use of bacteriophages, or viruses that attack bacteria, to determine similarities among different bacteria. Here you can see a figure that illustrates the phage typing process. Here you can see that receptor sites for bacteriophages are highly specific, and certain strains of a species of bacterium are attacked only by particular types of phage. Clear sites, or the plaques, are left when phages have killed the bacterial cells. On the basis of which phages have attacked a bacterial culture, one can determine which strain of that bacterial species is present. Here's a summary of some of the different classification criteria for bacteria. Now we know that, again, most macroscopic organisms can preliminarily be classified according to observable structural characteristics. However, it's more difficult to classify microscopic organisms, especially bacteria, because many of them have similar structures. So separating them according to cell shape, size, and arrangement does not produce a very useful classification system, nor does the presence of specific structures like flagella, endospores, or capsules. These do not allow for identification of a particular species either. So we must use other criteria as well. Some examples of these can include staining reactions, especially the Gram stain. We can also perform tests related to growth, nutritional requirements, physiology, biochemistry, genetics, and molecular analysis. These features include properties of DNA and proteins. And so by using these various classification criteria, we can identify an organism as belonging to a particular genus and species. You have an assignment in this class known as the unknown organism assignment. And in this assignment, you've been provided with a sheet full of information similar to that described in this table. And you will then use that information, those test results, to make use of keys like Berge's key, which we'll discuss in a moment, to determine the genus and species of your particular organism. Now, let's discuss Berge's manuals. Berge's manual is really the accepted reference on the identification of bacteria. This was originally published in the 1920s, and since then 8 editions have been revised and published. However, it's important to also recognize that Berge's manuals do not actually present an accurate picture of evolutionary relationships among bacteria. Instead, they're practical groupings of bacteria that make it easier to identify them. This manual is particularly useful as a reference and research tool. Now, for identification, there's also a Berge's manual of determinative bacteriology, and this one is actually based on a phylogenetic or evolutionary framework rather than on a non-evolutionary grouping by phenotype. Sequencing of 16S rDNA has provided guidance in this, but it's still very much a work in progress. Now, despite all of these taxonomic problems that we've discussed, there is an established nomenclature for bacteria. And bacteria nomenclature, again, refers to the naming of species according to internationally agreed upon rules. Both taxonomy and nomenclature are subject to change as new information arises. Organisms can sometimes be moved from one category to another, and their official names may even be changed. For example, the bacterium that causes tularemia, which is causes a fever acquired by handling infected rabbits, was for many years called Pastorella tularensis. However, its genus name was changed to Francisella after DNA hybridization studies revealed that hybridization between its DNA and that of Pastorella did not even occur. It does, however, have a 78% match with the DNA of Francisella. So when one considers the specific orders and families, we must remember that names have consistent endings. Orders always end in ales and families in ace. Now, as a beginning student, this may all seem quite confusing to remember some of the characteristics of the specific microorganism that we cover in the course. Also, Berge's manual itself is quite expensive. The 5-volume set is very heavy, and it's not something that you actually want to buy yourself. However, you can use some of the resources provided in the Blackboard. I've provided a key from Berge's that will be plenty for you to use as you identify your unknown organism. And you could also access the actual manuals themselves at the library at school. This is all we have for today. Thank you for your attention.